So, you knew at some point we were going to talk about the birds and the bees. Here it is. In this video, we're going to talk about meiosis. And meiosis is a special kind of cell division that makes sperm and eggs, which are your gametes. And those gametes are going to come together and make new individuals. There are going to be a few changes of note. One of them is because since we're planning on sperm and egg coming together, we really only want each of the gametes to have half the genome so that when the two halves come together, we have a whole. The other thing is that we really don't want all of our gametes to be exactly the same, because if all of your gametes are the same, then that means that all the individuals are the same. For example, you would be identical to your siblings. And if you're not a twin, and even if you are a twin, you probably don't want that. So this enables us to get a little bit more variation in the population. And we're going to talk about how those two things arise. Now, the first part of this process is going to look really familiar because it is basically mitosis. You are going to be creating a copy of the DNA and you're going to be dividing those cells. It's going to go through the exact same phases: prophase, metaphase, anaphase, telophase, and cytokinesis. And the same thing is going to happen during those phases. So during interphase, we're going to duplicate the DNA and get ready for division. In prophase, that chromatin is going to condense and the nuclear envelope is going to dissolve. During metaphase, those chromosomes are going to migrate towards the center of the cell. And towards anaphase, half of the material is going to go into each cell. To further understand what happens during meiosis, you need to remember that you have half of your DNA from your mom and half of your DNA from your dad. And what this means is if we were to look at a pair of chromosomes, one of those chromosomes is going to be from mom and one of those chromosomes is going to be from dad. A really, really cool thing happens between prophase and metaphase. Remember, during metaphase, those chromosomes are coming towards the center of the cell. When they are overlapping a little bit, they trade a piece of each other. And this trading a piece of each other is called crossover. And this crossover does something really interesting. It makes it so that when we reach our end point and we have new cells, those cells have a unique complement of genes. They're non-identical. And this is part of the big difference between meiosis and mitosis. Mitosis, you want to make identical cells. In meiosis, you don't. Now, it's important to mention here that these chromosomes are homologous. And what that means is that they're doing basically the same sort of function. So you're kind of just trading the mom gene for hair color for the dad gene for hair color. You're not changing genes that do very different things. However, sometimes this can lead to new combinations of genes, and that can lead to new traits in the population. And the exact location of where this crossover occurs is somewhat random. So this is another thing that's making it so that you can have many gametes, and the likelihood of having any that are identical is very, very low because these crossover events have a lot of different places that they can happen. So the thing about meiosis, or another difference, is that we go through it twice. So when we get to the end of the first meiosis, we're going to have these two cells, but these have the same amount of DNA as the parent cell. And what we're going to do is we're going to actually go through another division. This is after a short sort of what's like an interphase, but it's called interkinesis. And during this phase, there's no DNA duplication. So what's going to happen is we're going to end up with those chromosomes moving into a prophase where they're going to condense again if they decondensed. And now those microtubule organizing centers, or the centrosomes, are going to go to opposite poles. Here they will attach at the kinetochore of each of these sister chromatids And then during metaphase, they'll get those sister chromatids, or those chromosomes, aligned in the center of the cell. Now, here I'm just showing one chromosome, so I can't really do that. But when we go on to anaphase, what's going to happen is those sister chromatids are going to get pulled apart. So now you're going to have basically one of the sister chromatids at each end of your cell. So at the end of anaphase, each of our two cells that we had now has its sister chromatids split apart and in opposite ends of its cell. So then we're going to undergo telophase and cytokinesis. And remember that telophase is kind of when the DNA is finishing up its dividing, and cytokinesis is the cell membrane dividing. And what we end up with is each of our cells, because we had that crossover, has a unique set of genes. So none of them look the same. And when we compare them back to the parent, you'll see that the parent here has twice as much DNA as these gametes. What we refer to this as is being haploid. And having all the DNA would be diploid. One of the terms that your book uses is they talk about ploidy, and that basically means how many of those identical chromosomes do you have. If you've got a pair, you are diploid. If you've got a single chromosome, you'd be haploid. In this figure from your book, you'll see that you go through basically all of those divisions twice. So notice that the first set is called meiosis 1, and the second set is called meiosis 2. So if we figure out the key differences between mitosis and meiosis, the first one is that during mitosis, you create two identical daughter cells, meaning those cells look exactly like the original cell. In meiosis, you create 4 unique daughter cells. So those cells have half the DNA of the original cell, and none of them are alike. That's because of that crossover event, which is another thing that happens in meiosis, but not in mitosis. If we look at it closer, these are sort of symptomatic of other differences that happen between mitosis and meiosis. This different number of daughter cells is because we have 2 divisions in meiosis, and we only do 1 round of divisions in mitosis. The crossover event is related to the fact that we pair homologous chromosomes in meiosis, and we don't do that in mitosis. And when we pair them, that gives them the chance to cross over. So these differences kind of are all related, but the key thing is that they're creating more cells, and those cells are unique and they have half the DNA, because someday they're going to combine with another cell with half the DNA and make a new and unique individual.